Hello friends, welcome to Sam online class. Friends, today we shall discuss about the tertiary structure of protein. In the previous video, we came to know about various protein secondary structure like alpha helix, beta pleated sheet, random coil, gamma helix and beta tone. If we will assume that the protein structure include only the secondary structural element, no additional level of organization is involved, then how the globular proteins will be spherical? The spherical shape of the globular protein indicate that various secondary structural elements like alpha helix and beta pleated sheet are further folded so that a compact structure like globular protein is formed. So tertiary structure refers to the folding of the secondary structural element in a specific manner so that a compact globular structure can be formed. The tertiary structure of protein also refers to the spatial arrangement of amino acids that are far apart in the polypeptide chain. As an example, today we shall discuss about the tertiary structure of a hemoprotein known as myoglobin. Myoglobin is a relatively small oxygen binding hemoprotein present in the muscle cell. The term myoglobin is derived from two Greek words. Myo means muscle, globin means it is a type of protein. Myoglobin is a conjugated protein. So it consists of a protein part and a non-protein part. The protein part is a single peptide chain that contains 153 amino acids, and the non-protein part is a iron containing prosthetic group known as heme. Because of the presence of heme, this protein is called as hemoprotein. Myoglobin is especially abundant in the muscle of diving mammals like seal, whale and porpoise. The main function of myoglobin is to store oxygen. So by having abundant myoglobin in their muscles, the diving mammals like seal, whale and porpoise stay submerged underwater for a long time. The molecular weight of myoglobin is 16,700 and its overall molecular dimension. The overall molecular dimension of myoglobin is 45 into 35 into 25 angstrom. The deep red-brown color of myoglobin is due to the presence of heme in it. Friends, this diagram explains the tertiary structure of myoglobin. The backbone of myoglobin consists of almost straight 8 alpha helical segments. These are the alpha helical segments. The 8 segments are designated from the end terminals as A through H. And these 8 segments are right-handed. The outline of tertiary structure of myoglobin is sausage-like, and out of 8 alpha helical segment, the longest alpha helical segment contains 23 amino acids, and the shortest segment contains only 7 amino acids. In addition to 8 alpha helical segments, there are 5 non-helical regions. The non-helical regions are present between alpha helical segments. Example is CD is a non-helical region that is present between the alpha helical segment C and the segment D. In addition to this, there are two additional non-helical regions. The two additional non-helical regions include two amino acids that are present at the N-terminus and 5 amino acids that are present at the C-terminus. In myoglobin, beta-pitted sheet is entirely absent, and this is the C-terminus. C represents the C-terminus and this is the N-terminus. The C-terminus fragment or the C-terminus peptide contain 5 amino acids and the N-terminus segment contain only 2 amino acids. So 8 alpha helical segments are folded in a specific manner so that the compact structure of myoglobin is formed with the help of various types of non-covalent bonds like hydrogen bond, ionic bond, hydrophobic bond and so on. Out of 153 amino acids that are present in the globin chain of myoglobin, about 79% of the amino acid, that is about 121 amino acids, are distributed in the alpha helical segments, whereas the remaining amino acids are dispersed throughout the non-helical areas. The myoglobin molecule is very compact. There is a very little space in the interior so as to accommodate 4 molecules of water. All polar R groups except 2 are present on the surface where they are hydrated, and most of the hydrophobic R groups are present in the interior of the myoglobin. All peptide bonds that are present in the globin chain are planar, all have trans configuration, and the heme, which is an iron containing prosthetic group, rests in the crevice in the molecule. At the center of heme there is an iron atom. The iron atom, which is present at the center of heme, has 6 coordination sites. Out of 6 coordination sites, 4 coordination sites are occupied by the 4 nitrogens of the 4 chiral ring. Remaining, there are 2 coordination sites. Out of this site, 1 coordination site is attached with the R group of histidine, and remaining site is attached to oxygen molecule. Upon oxygen, the iron atom descends into the plane of the ring, and after that the oxygen that is bound to the heme is stabilized by hydrogen bonding with the imidazole group of histidine residue. The myoglobin that is associated with oxygen is known as oxymyoglobin, and the myoglobin without oxygen is known as deoxymyoglobin. The interior of myoglobin contains almost non-polar residues like leucine, valine, methionine and phenylalanine, whereas polar amino acids are absent from the interior. So friends, this is about the protein tertiary structure, and as an example we discuss the structure of myoglobin. In the next video we shall discuss about protein quaternary structure, and we will take the example of another hemoprotein known as hemoglobin. For more updates, please subscribe my YouTube channel Sam Online Class.